we have one singular goal, and that is to deliver first-in-class therapeutics for Parkinson's disease. I'll spend about 2 minutes telling you, since I have so much time, I'll spend 2 minutes telling you about how Parkinson's is a great unmet need and how we have promising targets and effective compounds that modulate those targets, and that we have the team to be able to deliver these therapeutics. And then the last 2 minutes, I'll show you our computational platform that we use to come up with these promising targets. Since this is new tech, I thought I would focus on the technology a little bit more. So 10 million people worldwide with Parkinson's disease. The best therapeutic we have is from the 1960s, and it only masks the symptoms of the disease. There is nothing that modifies the disease. So I really don't need to make leaps and bounds as to why this is a great unmet need. But 90% or more of the pharma companies are looking for acquiring deals, acquiring therapeutics that are working on CNS, particularly diseases like Alzheimer's disease or Parkinson's disease. And there has been a shift in focus for big pharma companies. Instead of doing these early stage discoveries in-house, they're preferring to purchase them from biotech companies like us, which is a great opportunity for us. So this is what our roadmap looks like. We're actually quite a bit of the way through. We have we've identified great targets that we think, biological targets that we think are very important in Parkinson's disease. We have compounds that modulate these targets, and we're in the stage of now confirming these compounds and coming up with a lead compound that we can take all the way to the clinic. This is looking like about 3.5 years from right now, and we actually just formed the company this week, so it's brand new. We're not raising funds yet, but we will be in September. So when we raised funds for the parent company, it only took us a month to open and close it. So if you're interested, let me know now before we open the round. But at the IND stage, we are planning on partnering with a pharma company to take it into human clinical trial. This is our team. My background is in neuroscience. I have a PhD in neuroscience and 15 years of experience in Parkinson's research. My co-founder, Andy Lee, who's sitting right there, was previously VP of technology at a Fortune 500 company. We pride ourselves in bringing technology and science together, and we also pride ourselves in bringing innovation and experience together. So a couple of the people on our team, Dr. Merchant and Dr. Romero, are veterans of industry, both at Eli Lilly and at Pfizer, bringing in over 25 years of experience each in drug development. So I'll tell you a little bit about the software now, which is the fun part. This is a screenshot of the software. And what it does is we create a virtual three-dimensional space in the computer simulation. And within that, we can put in a cell. So this is a neuron, a picture of a neuron, has a cell body, an axon. And I have highlighted the axon terminal region where you can see it kind of blown up here on the screen. And within that, we instantiate organelles such as mitochondria or lysosomes. And within that, even you can have intra-organelle compartments. And we put in the various different biological entities that are normally present in the cell, like proteins, lipids, and various small molecules. And we give them properties that they would ordinarily have in life, like mass and size. And because we give them these properties, we can actually press play and move them with time according to Newtonian laws of physics. So basically what we've done is created a virtual cell. And why this is important is because it allows us to create models of disease, so we can give this a genetic mutation, and then we can watch what abnormalities happen with this genetic mutation that causes Parkinson's disease, for example. And while we work on Parkinson's disease, this simulation can be used for any type of disease. Once we've created a model of Parkinson's disease and see these abnormalities, then we can add in compounds and see if we are able to reverse some or all of the abnormalities that we see in the disease model. We've spent 2 years now extensively validating this platform. This was funded by the Michael J. Fox Foundation for Parkinson's disease and in collaboration with the Mayo Clinic, who did the validation in vivo. And I'd be happy to talk about this more if there are questions. The data that feeds into the system falls primarily into 3 categories. I know there are a lot of data geeks out, so if you're interested in the data, I'd be happy to talk about that more as well. But primarily 3 categories. There's the entity information, like mass and size that I talked about, and also amino acid sequence, and those types of things. We use tons of omics data, so transcriptomic and proteomic data, and that tells us what to put inside each of these cells, particularly for a disease like Parkinson's disease that doesn't affect all of your cells in the same way. This becomes really important as to what type of cells we use. And then, of course, the biological interaction rules come from various different public databases, and these are kind of the most challenging things. And then, of course, the simulation is very resource intensive. So we run these on the cloud. I think at the highest we were running these on about 100,000 cores. And since we are at Microsoft, I'll give them a shout out. We were part of their BizSpark program, and they sponsored 2 years of research on this. And these are running on NVIDIA GPUs. I'll be happy to take questions. Yeah, actually, I mean, we designed with the blood-brain barrier in mind. The question was, how are you tackling the blood-brain barrier? Because this is a CNS disorder. So we're designing with that in mind. So we use a lot of computational chemistry tools, and we're, the starting molecules that we have all have the proper lipophilicity and the proper size to be able to cross the blood-brain barrier. We are working with small molecules, so it's a bit easier than, some of the other approaches that are going on. And a lot of advancement has actually been made on that front. But yeah, I mean, computational chemistry is primarily how we're going around that. Yeah. Yeah, really good question. So there is actually no real in vivo model of Parkinson's disease, but the laboratory model that we used with Mayo Clinic was almost identical to what we used, except we tried to use as much human information as possible in the simulation. Theirs was the exact same genetic mutation, but in mice, and it was a knock-in model, so they expressed it in the right amounts in the right places. But we tried to mimic exactly human. So all of the data that we use has been taken from humans. And one of the things that we're working on now is looking at biomarkers of disease, and that is something that we can actually test in humans. Unlike, obviously we can't do it in humans until we actually do the trial in humans. But as far as the animal model, we went as close as we possibly could with the model, with using the exact same genetic mutation in the same amounts. Thank you.